Hi, thanks for the invite. Thank you to Sharon. I will just share my screen. Well, it's a pleasure to spend a few minutes speaking with you. I will be making a little bit of a switch from a very practical presentation to a more conceptual and I would say hopeful presentation, because my lab at Mass General is interested in and focused on developing new therapeutics for NF disease. The therapeutic strategy I'm going to talk about today is not yet in clinical trials, but we hope to be there before long, as I'll allude to. I'll also mention that I'll be showing you quite a bit of data, but really only for conceptual reasons. And I'm not going to be talking through it in a highly detailed manner, but I did want to put it up for those of you in the audience who have the background that would make you interested. So I do need to disclose that I'm a founder with Equity. I'm also the chair of the clinical advisory board of a company called Mulberry Biotherapeutics. Their entire mission is to bring bacterial therapy to NF2 disease. As I'm also an inventor with patents pending related to the technology that I'll talk about today. Basically, everything I'm going to tell you about is available online in this PNAS paper, which is open access, which means we pay the journal some extra money and they let you read it without making you, buy the publication. So you can go, and if you're interested, you can read this paper and get a little more information. So schwannomas, which are the therapeutics that I'm initially interested in developing a treatment for, though, as you'll hear later, I would expand it to NF1 tumors, and we've been working on that. They're non-malignant tumors that grow on peripheral nerves, but as you may be aware, peripheral nerves are also within the cranial vault, associated with the brain, as well as associated with the spine. So while they're not cancers, and they virtually never become cancers unless you expose them to radiation, they do a great deal of harm to patients, as you may be aware. They appear first typically in children and young adults, with new lesions forming throughout life. So, and I'm really talking mostly about NF2 and schwannomatosis. Multiple tumors are not only frequently present, but really for NF2 and schwannomatosis, they're always present. With sporadic schwannoma, you can have one tumor. And they're really debilitating, as you guys are all aware. And there's a variety of neurologic problems, including deafness, balance issues that can cause blindness. And I'm a pain doc, which is how I really got interested in working towards a treatment for this disease, because I see NF1 and NF2 and schwannomatosis patients in my pain clinic. And very sadly, there's very, very little we have to offer these individuals for the most part in terms of pain control. There's substantial psychological comorbidity, both for the patients and for their families, for their loved ones. And the current standard of care, which is mostly operative resection, is associated with its own significant morbidity. And at times it's not even feasible because of the location or number of tumors. And you can see images that are just highlighting kind of what these schwannomas schematically look like. They grow from actually cells that are associated with peripheral nerves, not the nerve cells themselves, and often kind of they form a solid tumor that can kind of push the nerve bundles out of the way. And they're growing from these cells that myelinate axons called Schwann cells. And this is just a picture of a peripheral schwannoma associated with peripheral nerve. NF2, which, as you heard up front, is now called NF2-related schwannomatosis, or NF2, and formerly known as neurofibromatosis type 2. Just a little more information on NF2. It's due to a mutation of the NF2 gene, which produces the protein myelin, and that has a variety of effects on the cell. 50% of individuals with NF2 have a family history, so it's passed on, but 50% are spontaneous with no known family members with the disease. And the hallmark of NF2 is bilateral vestibular schwannomas. The vestibular nerve is one of the cranial nerves that run along the brain, obviously, and it's a hearing nerve. Multiple tumors are always present, and again, I would just remind you, they're intracranial, spinal, and peripheral, and they all can really create tremendous harm to patients. For example, down here you see an image of a cervical schwannoma pushing on the spinal cord up in the cervical region, which is really catastrophic. And it's just to remind you that while they're peripheral nerves, there's peripheral nerves in the spinal canal as well as in the cranial vault. This is also to images just let you know that NF2 disease is also associated with meningioma. About 50% of individuals will have meningiomas, which are brain tumors, and they can be benign or non-malignant or malignant, as well as a tumor called an ependymoma, and about 30% of individuals have ependymomas. So, as most of you are aware, it's a really, really bad disease entity. Median survival with NF2 is approximately 15 years after diagnosis. We don't know as much about the natural history as we'd like to, but this is some of the data that we have. And there's an indication that the earlier you're diagnosed, the more severe the disease is. I think whether that's really the case, we'll find out as time goes by. Obviously, we're diagnosing earlier and more frequently now than we did in the past. Just to get some idea of numbers, prevalence in the United States of NF1 and these schwannoma-associated diseases. This isn't really the audience for this, but I like to let people know that neurofibromatosis represents at least 3 or 4 distinct disease entities, right? And NF1 is genetically completely different from NF2 and the other schwannoma-associated diseases. NF1 is the most prevalent with about 100,000 US individuals, and it's due to a defect in the NF1 gene. And you know that there's over 1,000 known mutations that cause disease, and they cause neurofibromas as well as other issues that you're well aware of. Schwannomas are found with sporadic schwannoma. Usually people have a tumor that becomes clinically significant, and then when it's one tumor, frequently it can be removed surgically and it's not a big problem. We think that the prevalence is about 35,000 US individuals. Some of the people who are thought to have sporadic schwannoma turn out to have NF2 or schwannomatosis. The tumor was just caught very early. NF2, I mentioned Schwann lineage cells. All the tumor cells are Schwann lineage cells, as opposed to NF1 tumors where it's a mixed cell type tumor. Though having said that about NF2 tumors, the tumor, as you'll hear in a moment, also has many immune cells that contribute to its growth, most likely. It's due to the NF2 gene mutation, it's autosomal dominant, and there are cranial nerve 8 tumors always. And then there's schwannomatosis, which is the most rare of these schwannoma-associated diseases, probably about 6,000 individuals. Again, all the tumor cells are Schwann lineage cells, but it's a completely different genetic basis from NF2 disease. So the kind of focus of my talk is development of this bacterial therapy for NF2. And I will mention we've developed some gene therapy strategies, but that's beyond the scope of this talk, given time limits. What's the rationale? There's a very large body of clinical and preclinical evidence that supports bacterial cancer therapy. And I'll mention that an attenuated bacterium is the standard of care for bladder cancer before it gets into the muscular wall. And this therapy, which is readily available, cures 60% of people. Bacterial cancer therapy, the bacteria directly enter and can kill tumor cells. They inhibit angiogenesis or vascular formation around the tumors, which is necessary for tumor growth and survival. And importantly, they can cause what's called immunogenic cell death, which is killing tumor cells in a way that kind of alerts the host or the individual with the tumor that, hey, there are tumor cells here, kind of attack them. Before our publication, there were no reports of bacterial therapy for non-cancerous or benign neoplasms. And I think that was a major oversight. And I do have to thank my coworker in my lab, my former postdoc, and he's now an instructor, for bringing this whole idea of bacterial cancer therapy to my attention. And once we discussed it, we really got excited for reasons beyond the scope of this talk, and we pursued it aggressively. Why does bacterial therapy make sense for schwannoma? Schwannomas are solid tumors with hypoxic or low oxygen cores, and these bacteria that we're going to use, they love low oxygen environments. That's where they thrive. They're slow growing, and we've hypothesized that that would make bacterial therapy work better in these tumors than cancers. And they're highly vascular, so inhibiting vascular formation should help inhibit tumor growth. And as many of you know, bevacizumab or Avastin is one of the few treatments right now, pharmacologic treatment or drug treatments, that has any value for schwannoma, and that's an angiogenesis inhibitor. And I will also mention that schwannomas are what we call immunologically cold, or it's what we call a suppressed immune environment, where for a variety of factors that aren't fully understood, that kind of like signal to the host immune system that says, hey, I've got tumors here, I got a dangerous thing, kill it, gets suppressed. And bacteria have the ability to reverse that. There's a lot of data for that. And that's what I just mentioned. These tumors are infiltrated by a variety, not just macrophages and lymphocytes, but also natural killer cells and other white cells of the immune system, but they're in a suppressed state, we believe. So what did we choose to use? Of many possibilities for bacteria, we chose attenuated Salmonella typhimurium. It's not a natural pathogen for humans. It's a gram-negative bacteria that thrives in low oxygen environments. As I said, that exists in these schwannomas. It's genetically modified to minimize the potential to cause harm to human beings, and also modified to be even more dependent on the tumor environment for survival. The second part was done so you could inject it intravenously. So these bugs actually have a way to get out of vessels and into tumors. They're pretty amazing. And then only the ones that get into the tumor can survive. There have been multiple clinical trials using this specific strain, VNP20009, used intravenously, intratumorally, and even orally, that shows that you can use these bacteria without substantial toxicity. And they are anti-angiogenic, they block vessel formation, and pro-inflammatory. they can activate the immune system. I won't go into this too much, but I'll let you know there were no animal models, really, no good animal models when we came on the scene. So we developed 2 animal models where we implant human schwannoma cells into mice that have no immune system, really. They have no specific immune system. If they did, they'd kill the human tumor cells. Also, a mouse schwannoma model where we can implant schwannoma tumor cells into a mouse with a normal, relatively normal immune system. And that's important to look at immune effects. So we can implant, we can use special technology to follow tumor growth without having to open up or kill the animal. So we can treat with bacteria and follow growth and see what happens. And this was our first experiment, and we were just astounded. And what you see here is we actually modify the tumor cell so we can add a ligand substrate, we call it, into the animal, and the tumor cells actually light up, and we use a very sensitive camera. We can see the light. So this is why it says total flux or photons. And here you have PBS, which is just saline solution, that's a control, or the bacteria. And for a variety of reasons beyond the scope of this talk, we chose to use direct injection of the bacteria into the tumors rather than IV administration. But we get these tumors growing, and then we inject tumors either with saline or salt solution, which is the control, and that's the black bar, or with the bacteria, and that's the green bar. And you see, if you just inject them with saline, they just keep growing and growing and growing over time. If you inject them with bacteria, they actually resolve. And we've repeated this many times now, and every time we see the same thing. The untreated tumors grow, the treated tumors, most of them resolve. 75% of the bacterial injected tumors fully resolve. We also repeated this finding in that was in the human tumor line. This is, sorry, I didn't mention this is all with the human tumor schwannomas. Then we looked at mouse schwannomas. They grow a lot faster, but you see essentially the same effect. Untreated tumors grow and grow and grow. You actually have to kill the animals because they get so big. The treated tumors are controlled. What are the mechanisms that killing occurs by? And I'm going to go through data fast. There's something called apoptosis or programmed cell death. We believe this is when the tumor cells are invaded by bacteria. It leads to this programmed cell death. It occurs in both the human tumors and the mouse tumors. Everything from here on in is just the mouse tumors. We also showed there's inhibition of vascular formation, and these are the salt-treated tumors. Trust me, they're huge and they're full of vessels. And in the bacterial-treated tumors, all this is just like basically pus and fibrous material, and there's essentially no vessels left. We also saw increase in the number of immune cells, both T cells and macrophages. And amazingly, we saw a change in the type of macrophages from M2 macrophages, which are thought to actually help tumors grow, to M1 macrophages, which are pro-immunologic, anti-tumor or tumor killing. So it's kind of everything we had hoped to see. Amazingly, again, we see an increase in killer T cells with bacterial treatment. And critically, we see a decrease in something called Tregs, or regulatory T cells, which are the key cells that create a cold or immunosuppressed tumor environment where the immune cells don't recognize or are deactivated in terms of tumor killing. So amazingly, we saw everything we'd want to see. Now, we're not geniuses. A lot of these findings have been seen in cancer models, but never in a model of a benign neoplasm. And I'm going to run through this very quickly, but we asked the question, what would happen if an animal had two tumors and you only injected one of them? Because we saw this immune activation, and what we were hoping and hypothesizing was you could activate the immune system to kill tumor cells, not just in the tumor you injected with bacteria, but in another distal tumor, because these immune cells all circulate, of course. And what I'll tell you up front is when you inject one tumor with bacteria, the bacteria do not make it to the second tumor. And I'm not going to talk to you about checkpoint inhibition for time today, but let me tell you, it does help. And most cancer treatments aren't one modality. That's why we look at checkpoint inhibition. But if you treat with bacteria, again, it's the green line, you see that significant suppression of tumor growth compared to just saline. And this is in the injected tumor. And this is the amazing finding. In the uninjected tumor, so the tumor that's not treated with bacteria, here's saline, the tumor grows and grows and grows. And this is what we don't fully understand. But the non-injected tumor in the animal where the tumor on the other side got bacteria is also controlled. And it seems to be controlled even more than in the bacterial injected tumor. We're really thinking about why that would be, but it's an amazing finding. And it raises the possibility that we could treat one tumor in a human being and suppress the growth of the other tumors that are present at the same time. We always do something called a re-challenge model. And what we do is we implant a tumor, we treat it with bacteria, we wait, and then we implant another tumor and see what happens. And that's to ask the question, is there memory, immunologic memory, whereby once you treat one tumor, might it suppress tumors in the future? And I'll just show you. We repeated that finding again, where if you don't treat the tumor, it just keeps growing. But if you do treat it with bacteria, it's suppressed, albeit not completely. These tumors are growing quite fast. And again, we had this amazing finding. If we wait well after we treated this one tumor and try to implant again, there's almost no growth of tumor cells. So there is an immunologic memory response that can suppress the development of tumors that arise later in time. Quite an amazing finding. I will also mention that we showed efficacy of bacterial treatment in both NF1 deficient and proficient. So a malignant peripheral nerve sheath tumor model, which is NF1 associated type of tumor. And we see clear efficacy. And also with meningiomas, both benign and malignant, because we want to try to expand this to other tumor types that affect NF2 and NF1 and schwannomatosis affected individuals. So a little bit of a summary. I'm just about at half an hour, 25 minutes. We've demonstrated very clear efficacy of bacterial therapy in two distinct NF2 schwannoma models. A xenograft, that's not important, but it means when you take human cells and put it into another organism, in this case, mice, human schwannoma model, as well as an allograft, which means you're using cell type from that animal, mouse schwannoma model. Very clear efficacy. We've defined mechanisms of action, tumor killing via programmed cell death or apoptosis, inhibition of the blood supply via anti-angiogenic effects, shift to tumor killing macrophages, increase tumor killing and decrease suppressive T cells, and generation of an immunologic cell death, including, importantly, development of host anti-tumor immune responses that go throughout the animal, killing two tumors, even the non-injected one or treated, as well as killing tumors after initial treatment. We have shown, I showed you some evidence, initial evidence for using this approach or bacteriotherapy with other NF2, i.e. meningioma, and NF1 tumors. I have every reason to think this would work with neurofibromas, given what I know about the immunological environment. We haven't tested that yet. Our ongoing work, what do we really want to do? Move to clinical trials. We got very positive feedback from the FDA after submission of our pre-IND package, and we have also been awarded orphan status. We're working hard to develop even better or next generation bacterial strains to both enhance safety, which we've already done, just so you know, and improve efficacy or how well it works. I want to acknowledge several individuals. Sharif Ahmed, he was a postdoc at the time when he first brought this idea to me. And we really work well together. We've developed a variety of treatment strategies. John Mekelanos, who's a collaborator at Harvard Med School and just an amazing, amazing expert on bacteria. Xinhui Wang, I don't have a picture, but she's a tumor immunobiologist. I also actually have a PhD in immunology, which no doubt was partially why I went down this road. Manlin, it was a post-tech who helped. And I want to highlight the whole Mulberry Biotherapeutics group. Nothing gets to clinical trials really, or at least not quickly, without the support of industry. And I just want to give a shout out to Neil Kirby, the CEO, who's just amazing, amazing, amazing. We're fortunate to have him working on this project. We've had funding from NIH, NF2 Biosolutions, and I want to particularly thank Nicole Henwood and Gilles Atlin from that organization. They've provided quite a bit of funding. We wouldn't have been able to do this work without them, as well as my department for all their support. I think with that, I will just say thank you. I'm pretty much out of time. I hope that this was of some interest to the group. Thanks again for the invite.
Are there any questions in the room? If not, I have one for from online.
Thank you, Dr. Brenner. That was really eye-opening. I hadn't heard that before. That's absolutely amazing. Very exciting. We do have an individual that would like to know if you could say again how she could read the journal that you mentioned in the very beginning of your talk.
Sure. If you search on my name and PNAS or Proceedings of the National Academy of Sciences and kind of schwannoma, bacteriotherapy, if you put a few keywords in, that should come right up. If you give me 1 minute, I might be able to even pull up a link. Give me 1 second if you have it. Yeah.
Dr. Brenner, while you're pulling that up for us, another question came in and they're wanting to know if you have an estimated time frame to get to clinical trials.
Sure. People always ask that, and it's a very reasonable question. Realistically, it certainly wouldn't be less than a year. If we have adequate funding, and I think we still need to raise some capital down the road where we have monies now. Hopefully it would be no more than 2 years, but honestly, I really defer those kinds of questions to the company. I just put in a link because that frankly is a little outside of my scope of expertise in terms of translation at the industry level to clinical trials.
I think that's it on the questions. Thank you, Dr. Brenner, for your participation today. We really appreciate you giving your time here on a weekend for us to learn about your study. Thank you very much.
My pleasure. Thanks again for the invite.